So, role of enzyme in the regulation of metabolism. Before that, writing any things about role of enzymes in metabolic regulations, you must write first definitions. So, is a crucial maintaining the balance and efficiency of biochemical reactions, living organism. We are in plant scientist, so we are studying only a plant enzyme. So enzymes are a specialized protein that act as a catalyst and accelerate the rate of reactions without, without being consumed or used by themselves in process. They remain as such. So without being consumed in the process, they have a fundamental role in the regulating metabolic pathways by controlling the flow of substrate and product, ensuring that metabolic process occur at appropriate time and in correct quantities. So you can see this explanation of the various ways through which enzyme contribute the regulation in the metabolism. So first, enzyme activity control. How the enzyme activity can control or regulate. So enzyme can be regulated through the various mechanisms to control their activity level. One common method, as I said in the previous lecture, allosteric regulation. So these allosteric regulation are made up of two things. One is activators, another inhibitors. So where molecules bind to specific site on the enzyme other than the active site, it alters in the shape and consequently thereby affecting the activity of the enzymes. Allosteric regulators can either enhance or you can say allosteric activators or inhibit or you can say allosteric inhibitors. This regulation allows for fine tuning of metabolic pathways based on the two things. One is availability of substrate or need for the certain products. Now another, enzyme synthesis and degradation, that can also be regulated in the metabolisms. So the production as well as the degradation or closing of the enzyme are tightly regulated to maintain the optimum metabolic functions. These optimum metabolic function can be controlled through an inherited capacity. It means the plant or living organism have their genetic constituents. It means gene expression level. So gene expression control the synthesis of enzyme, ensuring that they are produced as and when they needed. Certain enzyme can be regulated through gene expression level. So another, in transcriptomics also, one of the term is transcription factors. So these transcription factors and regulatory elements on the DNA sequence that determines when and how much enzyme can synthesize in particular reactions. So additionally, enzyme can be degraded through various mechanisms. Which are the mechanisms through which the enzyme degraded? So one of the important mechanism, as you know that I have already explained in the factor affecting enzyme activity, such as proteolysis, to prevent their accumulation beyond their necessary level. Now, third, feedback inhibitions. That feedback inhibition I will explain in depth in next lecture. But here you have to write feedback inhibition is a common regulatory mechanism in which the end product of metabolic pathways act as an inhibitor for enzyme earlier in the pathways. I already explained in the glycolysis also. This negative feedback loop helps maintain homeostasis, equilibrium constant, by preventing excessive production of certain metabolites. When the concentration of particular product reaches at a certain threshold, it binds to allostatic site or allostatic inhibitors on the enzyme. It involved in the synthesis as well as in the inhibiting the further production of that particular product. So one of the regulatory mechanism in the metabolic pathways through enzyme is a feedback inhibitions. Fourth is covalent modifications. These covalent modifications, as you know that enzyme undergo a covalent modification such as phosphorylation, dephosphorylation, which can alter which can alter where I was. Yes, covalent modification. I have already told you there are two mechanisms. One is phosphorylation, another is dephosphorylation, which can alter their activity. And protein kinase and phosphate group to the enzyme while phosphates remove them. During the dephosphorylation or phosphorylation, what will happen? These activity can be altered proteinase kinase, add phosphate group to the enzyme, while phosphatase remove them. This modification can either activate or deactivate the enzyme, depending upon the specific metabolic context. Covalent modifications provide a rapid or reversible means of regulating enzyme activity in response to cellular signals, as I said in the competitive inhibitor and non-competitive inhibitors. So enzyme compartmentalizations is also localization within the specific cellular compartment or organelle, or it allows the special regulation of metabolic pathways by conforming the enzyme to specific region. So the efficiency and specificity of the reaction can be enhanced. For example, enzyme involved in the oxidative phosphorylations that are located in, as you know, that in the inner membranes of mitochondria, that ensuring the ATP productions. So this is includes enzyme substrate specificity, and I have already explained in the types of specificity. So 6 point is enzyme exhibit a high substrate specificity. Selectivity of the binding with the substrate and catalyze specific reaction. So this specificity allowed the precise control over metabolic pathways, thereby it ensuring that appropriate substrate act on appropriate enzyme. And enzyme recognize substrate based on their shape, charge distributions, and other molecular characterization, as I explained in the types of specificity. Now enzyme regulation through cofactor. So many cofactors, non-protein molecules called cofactors, and it has a functional properties. So cofactor can be inorganic ions, as you know that magnesium, zinc, organic molecules like as vitamins. The availability of these cofactor can also influence the enzyme activity. Therefore, by another ways, it regulates the metabolic pathways. So pyruvate, for example, if we see the enzyme pyruvate dehydrogenase, it requires a TPP, thymine pyrophosphate, a vitamin-derived cofactor for its activity. And last, enzyme regulation through pH and temperature. As I explained in the factor affecting enzyme activities, same thing is also involved in that regulation of enzyme. Have a optimum pH and temperature ranges. They exhibit maximum activity. For the optimal condition can affect the structure and functions of the leading to the decreased activity or denaturation of can be possible. So the pH and temperature cell can regulate the enzyme activity and modulate the metabolic pathways. So in nutshells, I can say that enzyme play a vital role in the regulations of the metabolism through various mechanisms such as enzyme activity control, synthesis and degradations, then feedback inhibition, covalent modification, compartmentalization, and substrate specificity, as well as O-factor regulation, including all factor affecting enzymes. So these regulation mechanisms ensure that metabolic pathways are finely tuned to meet the dynamic needs of living